2020 జేఈఈ మెయిన్ ఫలితాలలో NSR ఇంపల్స్ విద్యార్థుల ప్రభంజనం. ఆల్ ఇండియా అన్ని కేటగిరీస్లు 18, 35, 251, 253, 582, 606, 778, 780, 812, 821, 1031, 1049, 1303, 1575, 1654, 1753, 1754, 1978 ర్యాంకులే కాక మరెన్నో ఉత్తమ ఫలితాలు సాధించిన విద్యా సంస్థ NSR ఇంపల్స్. ఈ ఫలితాలు సాధించడానికి కారణమైన లెక్చరర్లు మరియు తల్లిదండ్రులకు మా కృతజ్ఞతలు. NSR ఇంపల్స్ ఎడ్యుకేషనల్ ఇన్స్టిట్యూషన్స్.
Hello students, now let us continue with the construction of the first recombinant DNA. Now we have discussed about what? We have discussed about the Salmonella typhimurium, right? So in Salmonella typhimurium, what we have studied? In Salmonella typhimurium, we are cutting and removing some part of the DNA, right? And that fragment has, this is nothing but the gene encoding antibiotic resistance. The gene resistance, antibiotic resistance. Gene encoding antibiotic resistance. The fragment of DNA what is extracted out, it shows resistance to antibiotic. Gene encoding antibiotic resistance. So when it takes the Salmonella typhimurium, it has the gene what is being extracted that shows antibiotic resistance. Now we cut it and removed. Does you think Salmonella further it shows a resistance to antibiotic? Definitely no. Okay, definitely it will not show resistance to antibiotic. Why? Because antibiotic resistance gene, the gene which is responsible for showing antibiotic resistance, we cut it and removed it out. Now Salmonella typhimurium further it will not show resistance to antibiotic because antibiotic resistance showing gene we cut it and removed it out. Okay, we cut it and removed it out. Now what we will do here? Now coming to this part here. This is E. coli. So E. coli now presently, let us take this is black one, this is nothing but alien DNA or foreign DNA we say. Now E. coli now no resistance to antibiotic, no resistance to antibiotic. Earlier no resistance to antibiotic. Now what happens here? Now with the help of plasmid, now this is acting as a vector so that this is get transferred into here, this is get transferred into here. How we are adding? We are adding with the help of DNA ligase. How we are cutting? We are cutting with the help of restriction endonuclease. Restriction endonuclease. We are cutting and extracted, and this particular fragment we are adding here with the help of another enzyme called DNA ligase. Now this will get transferred into this, and this plasmid will drop the foreign DNA, this foreign DNA into this, and the plasmid goes off and the plasmid goes out. Now this introduced plasmid what is being introduced here, now it will show earlier no resistance to antibiotic, now E. coli shows resistance to antibiotic. Now E. coli shows resistance to antibiotic. So what is introduced is nothing but gene encoding antibiotic resistance. The first enzyme used is nothing but restriction enzyme or restriction endonuclease, which is used for cutting off DNA. Okay, so I am just repeating because this is very important in biotechnology. Each and every step you have to understand. Restriction endonuclease involved in cutting of DNA and the fragment of DNA extracted out. This fragment of DNA added into the plasmid that act as vector. How it is added means with the help of another enzyme called as a DNA ligase. Now this DNA ligase is meant for for joining of DNA. Joining of DNA fragment. This is the second enzyme. Second, DNA ligase helps in joining of DNA fragment from here to here. Then with DNA ligase joins the plasmid with the foreign DNA or alien DNA. This is nothing but the gene encoding antibiotic resistance, and this get transferred into E. coli. Earlier E. coli no resistance to antibiotic. Then what is added here? It's called recombinant DNA. So this because of this recombinant DNA, now E. coli shows resistance to antibiotic. So this is nothing but the first constructed recombinant DNA, and they developed the technology called as recombinant DNA technology. What they developed, it's called recombinant DNA technology. What they developed is called recombinant DNA technology. Now, how this can be done? So now after they do this successfully, the transfer of the fragment of the gene into E. coli. E. coli here, it acts as a host cell, or it acts as what? Host organism, or itself it acts as a recipient cell. Host organism or itself is called recipient cell. Why do you call recipient? It is receiving this. Earlier E. coli no resistance to antibiotic, now it shows resistance to antibiotic. What get transferred and added itself is called new genome. It's recombined with the original genome. That's why it's called recombinant DNA, and with this Haber-Boyer-Cohen first constructed recombinant DNA, and they introduced the concept called as recombinant DNA technology. So recombinant DNA technology today it plays a vital role for transfer of the gene from one organism to other organism, and that is the basis for genetic engineering. So what they introduced now finally, what is very important one? Recombinant DNA technology. Now in this recombinant DNA technology, if you look at what are the main things here, there are three important steps. In this recombinant DNA technology, in recombinant DNA, what they first constructed, recombinant DNA, what is being constructed? What is that first constructed recombinant DNA? It is nothing but the gene encoding antibiotic resistance. Okay. Now for doing this recombinant DNA, for transfer of recombinant DNA, so for doing this recombinant DNA technology, basically they identified the process includes three steps. So for the first construction of recombinant DNA, when they have done that experiment, then they have understood for this particular process majorly it is divided into three processes, three steps. What is that first one? The first step is nothing but identification of desired gene on DNA. Identification of desired gene fragment or desired DNA fragment on DNA. That means what? So if you have taken the salmonella, so now first initially you have taken salmonella. In salmonella you have nucleoid, right? So in this nucleoid contain million number of genes. This nucleoid, this genetic material, what you find in Salmonella typhimurium, this genetic material or DNA itself contains millions of genes. So it has some long length of genes, it contains millions of genes. Out of this millions of genes, which genes show antibiotic resistance? That is the biggest thing which the people have to achieve by doing the experiments. There are some millions of genes on the Salmonella. Out of these millions of genes, some lakh number of genes, which fragment of genes show resistance to antibiotic? Which genes show antibiotic resistance? Suppose if I want to extract antibiotic resistance, which genes show antibiotic resistance is a question mark. So what I have to do? So genetic engineering theoretically to discuss on the board is so easy, but practically in the lab it may take centuries together to find the sequence of DNA from single cell. In that each gene represents what genetic character itself, takes lot of time. It takes huge time. It takes lot of time. Okay, so here when you take the here it takes little time, centuries together to identify which genes responsible for showing antibiotic resistance. So what is the first step we have taken here? Identification of desired gene fragment on DNA. Out of the million number of genes, which gene shows antibiotic resistance? That again they have to make DNA to cut into pieces with the help of restriction endonuclease, and they have to see which fragment contains what genes and they express what genetic characters. It takes lot duration of time. For the scientists, it is not an easy job and not an easy job to do it and to write on the board. It is very toughest and typical job, because they need to see the molecular level of the gene, and that gene when it is introduced into organism, what is a character is expressed by that particular gene. So everything they have to study and do research. It takes lot of time. It is a very typical and tedious process, not an easy job. It is a very typical and tedious process. Now in that first thing is identification of desired gene on DNA. Next one is introduction of desired DNA fragment into host. Introduction of desired DNA fragment into host. After identification of desired DNA, suppose this is a desired DNA what I identified, we need to cut with the help of restriction endonuclease, and then we have to introduce into host. That is the second step, right? And third step is maintenance of desired DNA fragment and transfer of this into progeny cells. Transfer of this into progeny cells. Progeny cells meaning what? Daughter cells. Transfer of this into progeny cells itself is nothing but what? A daughter cells. So here identification of desired gene fragment on DNA is the first step. Second one is introduction of desired DNA fragment into host organism, and then maintenance of that DNA fragment to be stabilized within the host, and make sure that when the host undergoes replication, this it should be get transferred into progeny cells or daughter cells. When host undergoes replication, along with the original genome, recombinant DNA also should get transferred into progeny cells. So these are the three important steps. They play a vital role, and maximum precautions we need to take, and when we are successful with these three steps, then we will be successful with the recombinant DNA technology. Then it is possible to inculcate recombinant DNA into host genome, thereby that expresses the desirable genetic characteristics. So these three are very important. What are they? Identification of desired gene fragment on DNA, introduction of desired DNA fragment into host, maintenance of desired DNA fragment and transfer of it into progeny cells or daughter cells. These three steps we need to take. Maximum experimental data it's needed, and which gene is showing resistance we need to do experiment. Transferring, it's not so easy job, we need to take precautions, and we need to do very carefully transforming into host cell, and we have to take precautions and much precautions and care until it get transferred into daughter cells to express the genetic character. Now let us continue here. When you take this here, so when we take this here, So when we take in this recombinant DNA, as I told you, this is not a very easy job. Okay. So what is that here we are doing? From the organism, this is nothing but the source organism. What we are doing? A fragment of DNA, we are cutting it, right, by the implication of what? Restriction endonuclease, restriction enzyme, right. A fragment is cutted with the help of restriction endonuclease. Let us imagine this is a fragment. Let us take, yeah. So what is extracted? It's called as what? Remember the terminologies carefully. Alien DNA, or itself is called foreign DNA, or itself is called as what? The DNA which is being extracted from the source organism. Okay. So when we take this alien DNA or foreign DNA, what is being extracted here? This is a very, very important and very first basic step, right. So when we take here the host source organism, what we find here, it's nothing but alien DNA, or itself we call foreign DNA. Now this, with the help of plasmid, we are introduced into plasmid. So this got introduced here, and with the help of another enzyme called DNA ligase. DNA ligase, it's for combining the alien DNA into vector, right. And finally what happens here? This will get transferred into another organism which act as the host organism, or the host organism itself we call it as a recipient cell. Itself we call it as what? A recipient cell. So in this case what happens here? Now this is a nucleoid, what we find here. Now the plasmid along with the alien DNA, it will come. It drops its alien DNA and plasmid goes out. Sometimes it stays, sometimes it goes out. Okay, when it stays, it's called cloning, and when it alone, it undergoes its own replication, cloning DNA. We have certain terminologies are there, you will come to know in the coming classes. If I tell you everything, it will be confused here. Sometimes the plasmid, it stays here, itself undergoes replication. But most of the times when used as a vector, it drops and it goes. So what do you call if it undergoes replication by itself without dropping, and what do you call if it leaves and goes out? That part we will discuss it later. We have a clear difference. Now in this case, when it drops and goes out of this place, when it drops here and goes out, let us imagine. So what is present here? Recombinant DNA is present, or what we find here is nothing but alien DNA. Now listen carefully, this particular alien DNA, what we find here, it will not show what happens here. It cannot replicate itself, it cannot undergo replication itself. Okay, so alien DNA cannot undergo, or it found that does not undergo replication, or it could not, or it don't have ability to undergo replication. Don't have ability to undergo replication, don't have ability to undergo replication. Alien DNA don't have ability to undergo replication. It was found unable to replicate. It was unable to replicate, or we can write the statement, it does not have ability to replicate, does not have ability to replicate, it does not have ability to replicate alone, does not have ability to replicate alone. All these statements are very important. You need to understand the statement very clearly. Alien DNA do not have ability to undergo replication, or you can say it was unable to replicate, or it does not have ability to replicate alone. What they thought initially, Herbert Breuer and Stanley Cohen, as soon as they dropped the DNA into this, then they dropped this DNA into this, then they thought that okay, it will undergo replication by combining with the genome, nuclear genome. But here it could not able to replicate. What they taught here, okay, we dropped the gene, fragment of DNA, it can undergo replication, They thought that simply dropping the gene, it will multiply and gives new genomic characteristics to the daughter cells. But later they found that it was not happening. Why? Because the fragment what is dropped into the host organism, into the recipient cell, it does not have ability to undergo replication. So they found that okay, when they observed initially in the beginning of the experiment, what I am telling you here, then they dropped into recipient cell and they waited for some time. They have done the experiment and they found that the progeny cells are having as it is nuclear genome with old characteristics, because this particular fragment was unable to replicate. When it is divided, it came into any one of the daughter cell. Other daughter cell is not having the recombinant DNA, and it could not able to produce a new characters. That means the dropped DNA fragment does not have ability to replicate. So initially they failed when they have done first time the experiment. Then later what they have done, when they took that, okay, this is not happening. In the very beginning of the first experiment when they saw, they thought it was not happening. So what they have done, next time when they have done second time experiment, now this time they introduce this DNA into nucleoid. That means alien DNA, or we call foreign DNA, integrated into genome, or you can write host genome or nucleoid, alien or foreign DNA integrated into host genome or nucleoid. When it got integrated here, again with the help of DNA ligase, in the next set of experiment they got introduced here. Alien DNA got integrated into this. As a result, now as a result what happened? Now foreign DNA become a part of the host genome of chromosome, and replicated along with host genome, into progeny cells. Okay, so what is that we are studying here? A foreign DNA became a part of the host genome of chromosome, so and undergone replicated along with the host genome and transferred into progeny. What is the meaning of this part here? So what is the meaning of this particular part here, what we are trying to discuss? Foreign DNA became part of the host genome. Now what happens here? After when got integrated into this, now this is now it is called recombinant DNA. Now this recombinant DNA became part of chromosome. It became the part of chromosome. When it become the part of the chromosome, it undergone replication, meaning it divides into two daughter cells. As a result, it produces two daughter cells. It produces two daughter cells, and two daughter cells has its own nucleoid, has its own genome, like this. So the progeny cells also has recombinant DNA. That means it could able to replicate along with the host genome. It became the part of, when it became the part of the host genome, it became part of the host chromosome, then it replicated, it got the ability to undergo replication along with the host genome, then it got introduced into progeny cells. Now this progeny cells, in this there is a change in phenotype as well as genotype. In the progeny cells, definitely we can see changes. Phenotype means morphologically, structurally, external characteristics or changes are there in external appearance as well as in genetic characteristics, because when it is alone outside, it don't have ability to multiply here. When got introduced into host genome, then it got ability to multiply, it multiplied along with the host genome. So this was very important discovery. In the very first time when they have done the experiment, they are not successful in making the multiplication of the host and recipient cell to produce daughter cells. It took long duration of time where they got multiplied and produced with daughter cells, or what are called as progeny cells. Right. Now this is how they discovered what is called as a recombinant DNA. Now let us continue with what are the tools of recombinant DNA technology. So what are the important tools? Tools means requirements. What are needed? Tools means requirements. What are needed for recombinant DNA technology? Let us make the list of the tools. So first thing is we need enzymes. Which enzymes? Let us take first one is restriction enzymes. Second one is a DNA ligase, and the third one is DNA polymerase. What are the important enzymes which we need? One is restriction enzyme, and other one is DNA ligase, and third one is what? DNA polymerase. Along with that we need a vector and a host organism. We need a vector and the host organism. So when we take the enzymes here, mainly we are in requirement of the three most important enzymes. One is restriction endonuclease, other one is ligase. Restriction enzyme is for cutting the DNA, DNA ligase is for joining the DNA, and for replication DNA polymerase. Yes or no? So what are the things they needed here? Restriction enzyme is meant for what? For cutting off DNA fragment. Restriction enzyme plays an important role for cutting off DNA fragment. And what for DNA ligase? For joining of DNA fragments. For joining of DNA fragments. What for DNA polymerase? For replication of DNA. DNA polymerase is needed for replication of DNA. Right, DNA ligase is for joining of DNA fragments, restriction enzyme is for cutting the DNA fragment, we need a vector to carry the fragment of DNA, and finally we need to introduce into the host organism to multiply and to produce the new genetic characters into progeny cells. So, tools of recombinant DNA technology, meaning what are the important requirements for recombinant DNA technology. The most important requirement, first enzymes. What enzymes? Restriction enzyme, DNA ligase, DNA polymerase. These three are nothing but enzymes. Along with that, vector and host are also needed. Let us discuss about the first one. Let us take on the tools. What is the very first one we have? Restriction enzymes. Restriction enzymes. So, when we take the restriction enzymes here, basically if you look at restriction enzymes, now first thing is, as I told you, every restriction enzyme has a specific site to cut the DNA. So, what do we say? A specific restriction enzyme needed to cut specific site of DNA. So, what is a very important aspect? Specific restriction enzyme, it is needed to cut a specific site of DNA. So, every fragment of DNA we have a specific restriction endonuclease. Initially in the year of 1972 they constructed first recombinant DNA, but in 1963 the people have identified, scientists identified two enzymes. Okay, in 1972 the first recombinant DNA is constructed. Before to that in 1963 the scientists identified the two enzymes. And these enzymes which restricted the growth of bacteriophage, in the host E. coli cell. Okay, what is the meaning of this? In 1963 they were observed there are occurrence of two enzymes, and these two enzymes they restricted the growth of bacteriophage in the host E. coli cell. What is the meaning of this? Restricted the growth of bacteriophage, meaning what? What is a bacteriophage? Phage means virus. Virus infected bacteria. Okay, what is that? Phage. Phage means virus. What is bacteriophage? I think you know it. In first year you studied virus infected bacteria. Virus infected bacteria. It's called bacteriophage. What do we call? Bacteriophage. Virus infected bacteria, it's called bacteriophage. Generally the viruses, they will attack the bacteria, they take shelter on the bacteria. I think first year biological classification we have studied, right? But there are two enzymes which are not allowing, restricted means what? Which are not allowing the bacteriophages to grow on the host organisms, which are not allowing the viral cells to grow on the host organisms. Then they try to identify what are these enzymes. They have entered into the depth of doing research work. Then they identified these two organisms as one of the enzyme, so I will just erase this statement. What is that? These two enzymes, guys, I will erase this point because I want to write the two enzymes down. So, try to make a note of it. These two enzymes, what they have discovered, restricted the growth of bacteriophage in the host E. coli cells. I will just erase that. So, here these two enzymes are nothing but one of the enzyme added methyl group to the DNA. Out of the two enzymes, one of the enzyme added methyl group to the DNA, named it as it was named it as methylase. It was named it as methylase. And another enzyme found that used in used in cutting of DNA. It was used in cutting of DNA. Then named it as restriction endonuclease. Named it as restriction endonuclease. What are the two enzymes? So, you can just make a note of it here. One of the enzyme added methyl group to the DNA, named it as methylase, right. Another enzyme which is involved in cutting of DNA, named it as restriction enzyme. What is the meaning of this? Now, if you look at, if this is a DNA strand, one of the enzyme is trying to add methyl group. So, it is trying to add what is called a methyl group. So, one of the enzyme is adding methyl group. So, that enzyme as is responsible for adding methyl group, so that is why it is called as what? Methylase. Alright, because methyl group when it is added, the DNA will be more stabilized, it gets more energy. And another enzyme, it was found when it is involved, it was found that the other enzyme involved in cutting of DNA, okay, and which is involved in cutting of DNA, this they have mentioned it as restriction enzyme. So, what are the two important enzymes they identified? One enzyme is adding methyl group at the terminal end, so the DNA will be more stabilized. So, that is called methylase. Another enzyme is responsible for cutting the DNA into fragments. They have given the name as restriction enzyme. So, one of the enzyme is methylase and other enzyme is what? Restriction endonuclease. So, when we take the tools of recombinant DNA technology, first important requirement is restriction enzymes. Then along with the restriction enzymes, what are the other enzymes which are needed here? The other enzymes which are needed here are nothing but we have DNA ligase, DNA polymerase, right, and these are three important enzymes, vector and host. Let us start our discussion with first most and foremost important requirement, that is nothing but restriction enzymes. That is, a specific restriction enzyme is needed to cut a specific site of DNA. So, this particular restriction enzymes, one enzyme we cannot apply for all. For every fragment of DNA, it has a specific site to cut it. So, I have finally people had identified, scientists identified two enzymes. One is, it was responsible for adding methyl group to DNA. It was named it as methylase. Other enzyme involved here, involved in cutting of DNA, it was named it as restriction endonuclease. Found that these two enzymes are restricting the growth of bacteriophages on E. coli.
ఇంటర్మీడియట్ విద్యా రంగంలో అగ్రగామి మన NSR ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి, జేఈఈ అయినా, నీట్ అయినా తిరుగులేని విద్యా సంస్థ NSR ఇంపల్స్.